All right, today we are going to start chapter 14 titled RNA and Protein Synthesis. This builds off of chapter 13, which was all about DNA. So remember way back in chapter 13, we talked a little bit about the history of DNA, how Griffith with his mice and the Monnier strands, there was a transforming principle, and then we went on to Avery, who confirmed that, hey, it's not protein because these enzymes destroy the transforming principle, destroy it. So it can't be proteins, it's got to be DNA. And then we had Hershey and Chase with bacteriophages. Then we talked about Maurice Wilkins and Rosalind Franklin with X-ray crystallography. Watson and Crick building models of DNA. And then we ended the chapter with how DNA replicates. Well, we're kind of going to pick up after DNA replicates. What else does DNA do? Remember, DNA's got 3 jobs. It's going to store information, it's got to copy that information, and then it's used in gene expression. So we're going to focus in on that gene expression. So today's objective, we're going to hone in on RNA, ribonucleic acid. What is it? How is it different from DNA? And how is RNA made? And then from there, we'll talk about how proteins are made, because proteins are essentially the reason why we are different from each other. Eye color. It's because different proteins were made for that different color, or skin color, or just everything. So DNA is the information molecule, but proteins are the ones that are really doing the work, that are the reason why organisms have variations. So here we go. Today's learning objective, describe how RNA differs from DNA. There's several different ways. And then how the cell makes RNA. Okay, so section 1 is about the role of RNA. RNA stands for ribonucleic acid. It's a single-stranded nucleic acid. And the sugar is ribose. So just in that one sentence, there are 2 differences. Single-stranded nucleic acid. Well, DNA was double-stranded. Contains the sugar ribose. Well, DNA was deoxyribose. So DNA contains instructions to build the proteins, but the thing is, DNA never leaves the nucleus. So how do ribosomes that are in the cytoplasm, that are on the rough endoplasmic reticulum, or the rough ER, how do they build proteins if they never get the instructions directly from DNA? Well, here's your answer. RNA. RNA is an intermediate link. It reads the DNA, is created in the nucleus, and has the ability to leave the nucleus to carry those instructions to ribosomes. So RNA serves as this intermediate link between DNA and protein synthesis. It's made up of 3 parts, very similar to DNA. Really, the only difference is the sugar, okay, ribose. DNA had deoxyribose. They both have a phosphate group, so the sugar and the phosphate kind of make up the backbone. And then we have nucleotides, okay, a nitrogen base. And we're going to discuss more about those nitrogen bases later on in this chapter. They're not exactly the same, but similar, okay? So differences between RNA and DNA. Ribose sugar versus deoxyribose sugar. RNA has 4 nucleotides, but it's not A, T, C, G. So remember, with DNA, A pairs with T, C pairs with G. With my little mnemonic device, all tigers can growl. Well, RNA doesn't have thymine. It has a different nucleotide called uracil. And so A pairs with U and C pairs with G. So my little saying for RNA is, all unicorns can giggle. Okay, another difference, single-stranded versus double-strand. And then finally, the last difference, and probably a very important one, short-term versus long-term. What I mean by that is that DNA is a long-term molecule. Yeah, your DNA can change through mutations and epigenetics and a few other factors, but for the most part, it's pretty stable and it doesn't break down. RNA, on the other hand, is a very short-term molecule. Sometimes the lifespan of these molecules could be minutes to hours to weeks to months, depending on the situation. So, like, for example, female mammals, when they give birth, they produce milk. And that milk is actually a signal from a protein called casein. So mRNA is carrying instructions, okay, to the mammary glands to say, hey, you need to secrete or make milk. And that protein is called casein. And so the casein is red, and it has a longer lifespan compared to a woman that is not breastfeeding. So that's just one example of short-term versus long-term. So the short-term can vary. So here are the nucleo bases or nitrogen bases for RNA here, cytosine, guanine, adenine, and uracil. There's thymine for DNA. You can see that it is a single ring, very similar. It's just that it doesn't have the CH group off to the side here. Mail time. Okay, so this chapter is all about the story of how DNA gets its instructions to ribosomes to make proteins. So we will talk about, well, we've already talked about how DNA can replicate, okay, when it's ready to divide. But we're going to discuss transcription today, how we get those instructions, okay, this piece of paper that's, could be destroyed very easily. How we create those instructions that can leave the nucleus, that can find a ribosome to become a protein. And then once it's done its job, once it's been translated, then it will be degraded and recycled. So RNA is this unstable short-term molecule. Now there are 3 types of RNA, okay? Messenger RNA, and that's the one that's going to carry the message from DNA to the cytoplasm to find a ribosome so it can become protein. There's ribosomal RNA, or rRNA as an abbreviation, that makes up ribosomes. So ribosomes make proteins. And then we have transfer RNA, or tRNA, and their main job is to carry amino acids to the ribosomes. So I'm just mentioning this, just know that there are different types of RNA out there, and we're going to talk about all 3 of these, but we're going to focus in on messenger RNA. And then when we talk about how proteins are made, we'll talk about ribosomal RNA and transfer RNA more in depth. So let's talk about how RNA is made, RNA synthesis. It is made through a process called transcription. Transcription is the process of copying a sequence of DNA to produce a complementary strand of RNA. It's very similar to DNA replication, except that we're not making DNA, we're going to make RNA. So here are the steps, okay? First off, RNA polymerase. Okay, RNA polymerase, not DNA polymerase like in DNA replication. RNA polymerase attaches to a promoter on a gene and the DNA unwinds. And then this RNA polymerase is only going to use one strand as a template to make a complementary strand. Because remember, mRNA, RNA here, is a single-stranded molecule. So there's no purpose to read the other strand. When it matches complementary, T is going to be replaced with U. So if there's an A on the DNA, then it will place a U to pair up with it. If there's a T on the DNA, it will place an A to pair up with it. So then the complete RNA strand, once it's done, will separate from the DNA and will leave the nucleus. So let's model this, right? So the first step is that RNA polymerase, is going to latch on, and the DNA is going to form like a little bubble so it can split. Okay, so I know this is kind of drastic, so maybe I'll just angle it a little bit. Nope, I don't want to do that. None of my nucleotides are angled. But let's just say, here's RNA polymerase, okay? Latches on and the DNA unwinds. Well, technically it would form a little bubble, but it unwinds, okay? And then we have these nucleotides, okay, floating around, and they're going to match complementary. RNA polymerase is going to help match it complementary. So my first nucleotide is G. G pairs with C. And then C pairs with G. Got another C here, so it's going to pair with G. A pairs with U, uracil. If it was DNA, then it would be thymine. G pairs with C. All right, I got 2 T's, so that means T pairs with A. Got another T. Where's my other A? A pairs with U. Sorry, that's going to bother me. and then C pairs with G. Okay, so I've made this single-stranded mRNA molecule, and that's going to fall off. And RNA polymerase is going to fall off as well, and DNA is going to zip right back up. Okay, boom, we read the gene that we wanted. This mRNA, maybe it's a gene, but it's going to go on and leave the nucleus and find a ribosome and hopefully become a functional protein that you need. So here's transcription, another diagram showing how DNA will just kind of unwind. One side does not get read, the other side does. Our nucleotides are matching complementary. A pairs with U, C pairs with G. All unicorns can giggle. And there's our RNA polymerase doing all the matching. So comparing transcription to replication, there are some similarities. Both occur in the nucleus of eukaryotes. They both use enzymes. They're both types of polymerases. Remember, ASE means it's a type of enzyme, and they do involve some type of complementary base pairing. There are some big differences, however. With replication, we're making a complete set of DNA. With transcription, we're only reading a small segment of that DNA, and there's a small segment of instructions. With DNA replication, we end up with a long-term storage. With transcription, the RNA that's made is a short-term storage. Going back to DNA replication, occurs once in each round of the cell cycle, and for transcription, it can happen as many times as the cell needs it to, so it's unlimited. So our next step or our next journey with this is what happens to the mRNA or the RNA that just got made in transcription. What happens to it after it leaves the nucleus? All right, so that does it for today. We described at least 4 differences between RNA and DNA, and then explained how the cell makes RNA with the steps.